Hey everyone, this is a continuation of my spring break reviews going over the chapter 2, or excuse me, exam 2 stuff. And this time we are on chapter 5, which is epistasis. So just well, I don't know what's happening here. Excuse me, sir. There we go. So epistasis, we're increasingly becoming aware in class that not everything is very simple Mendelian 9 to 3 to 3 to 1 crosses. We have things like penetrance that just kind of dominate the rules that we learned in Mendelian genetics. And one such example of this are the different kinds of epistasis. And so often in real organisms, many traits that we express are controlled or regulated by more than one gene. So we are not usually dealing with discrete genes, which are one gene coding for one function or phenotype. And epistasis is an example. An epistasis is where you have two genes interacting, and usually one masks or blocks the effect of another. One regulates another. And epistasis might seem like a funky word, but we can break it down. Oops. Epistasis. So epi, excuse me, means on top of. And so when we talk about two genes that are connected in the way of epistasis, there's one gene that is controlled by another, one that is masked by another. So we have, for example, a gene that's represented by the letters A, and then the second gene, B. A controls B, depending on whether or not A is capital or lowercase. It can mask the effect of B. And so because A controls B, A is like on top of B, and epi means on top of. So we have our epistatic gene and our hypostatic gene. Hypo is a prefix that means less than or underneath. So we have an epistatic gene and a hypostatic gene. And there are different cases that we study in genetics. One of these cases is recessive epistasis, arguably the most simple. So let's say, for example, that you have a flower, a certain kind of flower, and the color of its petals are controlled at the loci R and D. And if you have if you're dominant at the R locus, or actually, let me articulate that. The R locus, where is the pen, please? Oh no, what happened? Sorry about that. The R locus codes for color. And the D locus codes for whether or not color is deposited into the cells that make up the flower's petals. So R codes for color, D codes for whether or not it's deposited into the petals of the flower at all. And so if you are dominant at the R locus, you code for red flowers. And if you are recessive at the R locus, you code for purple flowers. If you are dominant at the deposition locus, you successfully deposit pigment. You successfully deposit color into the petals, but if you're recessive, you don't deposit color. And so we see that we have a red flower. It is, dominant at both loci. It has a dominant D allele, and because the deposition of color is dominant to not depositing color, we do deposit color successfully, and that color is red because we have a dominant allele at the R locus, the color locus. And conversely, we have a purple flower, which also successfully deposits pigment because of this dominant allele, but it has the purple color attribute because of its recessive nature, homozygous recessive nature at the color locus. However, we have a white flower here, and it doesn't matter whether or not it is dominant or recessive at the color locus, because no matter what, it is recessive at the deposition locus. So it doesn't matter what color is being coded for by its R locus, it's not going to deposit that color because it is homozygous recessive at this deposition locus. So we see that this gene, the deposition of color gene, is epistatic to, or it controls the effect that we see, or it controls the phenotype that we see of the color locus. So this is epistatic to this. And that's all well and good, but we can use this tendency and our knowledge of the previously defined Mendelian crosses that we're so familiar with to make up some problems or even solve some problems. And so we know in Mendelian genetics that if you have two true breeding individuals, let's use the same letters. So that's terrible. Let's say you have a true breeding homozygous dominant individual crossed with a true breeding heterozygous, or excuse me, homozygous recessive individual. I'm sorry, it's kind of late. So you cross these parents and you get what for your F1 generation? Well, obviously this individual can only give a capital R, only a capital D. This one only a lowercase and only a lowercase. So we have an R and a D capital, an R and a D lowercase. So all F1 individuals are going to have that genotype. And so what is this individual? Looking back at our loci, we have a capital D. We know that the capital D, or the D in general, controls what we see at the R. So let's always look at the D first, look at the epistatic gene first. So we have a capital D, which means that our color is going to be deposited. That's good. So now we can look at this and see what it's going to be. There is at least one uppercase R, which means it is going to be red. Actually, I might even write that in. No, I thought I clicked red. So this individual is red. That was very bold. And okay. So, and we have this individual. Let's look at the epistatic locus first, and we see that it is homozygous recessive at the deposition locus, which means no pigment is going to be deposited. So no pigment means that this individual is going to be white. It's going to have white petals. But if we look at the F1 generation, we once again look at the epistatic locus, and we see that there is at least one capital D, so pigment is going to be deposited. And now we can look at the other locus, and we see that the red allele is present, and that is dominant to the purple allele. So this flower is also going to be red. So 100% of the offspring of the parental generation of this true breeding, let me get my true, spelling. True breeding pair, 100% of their offspring are going to be heterozygous and red in the case of this example. So what happens if we cross two individuals of the F1 generation? We know this very well. It should end up being that beautiful, what we call a test. I cannot write today. Test cross. That was even worse than the first time. Forgive me. So we have that test cross. And that is a cross between two of these individuals. And we know from Mendelian genetics that it should end up being 9 to 3 to 3 to 1, where 9 individuals are homozygous at both loci. So, or excuse me, not homozygous, dominant at both loci, excuse me. And then 3 individuals are going to be dominant at the first locus, but recessive at the second locus. Another 3 are going to be recessive at the first locus, but dominant at the second. And then 1 individual is going to be homozygous recessive at both loci. This is what we'd expect from Mendelian genetics with discrete characteristics, but these characteristics are not discrete. They are epistatic, controlled or regulated one gene onto another. So what happened? Please give me the screen back. Thank you. Didn't even click the pen. So we have this test cross. And this, we can look at the phenotypes, or we can look at the genotypes and infer their phenotypes. So we have a capital D. We know that means that there is pigment, so we can look at this locus, and they are going to be red. And we can look at this locus. So we, right here, don't have any deposition of pigment. We have homozygous recessive at this second locus, the epistatic locus. And so we have white. Now onto these individuals, we have deposition of pigment and homozygous dominant at the hypostatic locus, which means these individuals are going to be purple. And once again, we have the no deposition of pigment, the homozygous recessive at the epistatic locus. So these individuals are going to be white. They don't deposit whatever pigment is made here in the hypostatic locus. Excuse me. And so we can see that 9 individuals are red, 3 are purple, and 4 are white. So we have a test cross from 2 true breeding individuals with recessive epistasis gives us a 9 to 3 to 4 ratio rather than the familiar 9 to 3 to 3 to 1 because of how one locus is epistatic over another. But recessive isn't the only kind of epistasis that we can see. Excuse me. For example, we have dominant epistasis. And so let's imagine that a snake can have a different kind of scale arrangement or state. And so it starts off bare. It just has its epidermis that hasn't secreted any hard scale parts. And if it has a dominant allele, then it is going to successfully, or if it has a dominant allele specifically at this S locus, it is going to code for the production of scales. Its skin is going to grow or build keratinous scales. However, that is only if there is a dominant allele at this S locus. So if there is a lowercase, or if you end up with a homozygous recessive genotype at this S locus, it is going to block the production of scales at all, because you need this capital S, this dominant S locus, or dominant allele at the S locus, to go from having bare skin to scaled skin. And conversely, if you have or you need a homozygous recessive genotype at the C locus to, instead of having regular colored scales, to have scales that help you camouflage, you need to have a homozygous recessive genotype at the C locus in order to have these camouflaging scales. So if you have a single, any kind of dominant allele at the C locus, that is going to keep you from having these, excuse me, sorry, my pen is acting up. Having any dominant allele at the C locus is going to prevent you from creating camouflaged scales. You'll obviously still make irregular scales, but that's only if you are able to get through this checkpoint. You get through this checkpoint by having a dominant allele at the S locus, and you get through this checkpoint at having homozygous recessive genotype, 2 recessive alleles at the C locus. That's what makes camouflage scales. So we can see that, any kind of, well, actually, with this dominant allele, it is, it can overpower a single recessive allele at this C locus. So this C locus is epistatic to this S locus, because obviously dominant alleles are, they dominate their recessive counterpart. And so it is more likely to have a pronounced effect than this locus. And so we see that, or we could, let's do a cross and make sense of the numbers. So we're expecting a 9 to 3 to 3 to 1. We know that. Mendel helped us find that. And so let's have a cross between true breeding individuals. And so let's say that we have an capital S, capital C crosses with a lowercase s, lowercase c. That is obviously the parental generation. It is going to give us a entirely or 100% frequent heterozygous individual at both loci. And when you cross 2 of these together, to create the F2 generation, we're going to have 9, 3, 3, 1. And so we have 9 individuals that are dominant at both loci, 3 that are dominant at one, recessive at the other, 3 that are recessive at one and dominant at the other, and then 1 that is recessive at both loci. And so we can see that these 9 individuals, I did this backward. That's embarrassing. Can I salvage it? Let's see. Yes, I can salvage it. Oh my God. Don't mind me. Do not look behind the curtain. Anyway, how's your day going? Oh God. It's like 10 o'clock. Anyway, so let's pretend that it was this way the entire time. And so we have a snake that starts with bare skin. And if it has a homozygous recessive genotype at the C locus, then it can move on to producing scales. And then if it also has a dominant genotype at the S locus, it can translate or convert regular scales into camouflage scales. So we can see here that having a capital, or excuse me, I keep confusing those, having a dominant allele at the C locus will automatically shut this process down. It'll stop us dead in our tracks right here. And any snake that has a capital, oh my God, dominant C allele will automatically be bare, regardless of whether or not they can, whether or not they have dominant alleles at the S locus, because we have the dominant alleles at the C locus that stop us in our tracks. We cannot move along this process of going from a bare snake to a camouflaged snake. And so we can look at this cross. And we see that we have, oh, right here, we have identified this to be the epistatic allele. It controls this locus, or not the epistatic allele, the epistatic gene that controls this gene, the S locus. And so we see right here, we look at the epistatic gene first, and we see that there's a capital C. So these 9 individuals are going to be bare snakes, just skin, no scales. But right here, we see, moving on, that we have the genotype required to move on with this process. So we have successfully built our scales and we can move on. So we have scales. Now we can look at the hypostatic locus and see if we can move on from regular scales to camouflage scales. And we have this necessary dominant allele. So we do. So this is a camouflaged snake. And we move on, but we see this epistatic allele, the epistatic dominant allele that prevents us from moving along the pathway. And so we have another grouping of bare snakes. And then obviously here we have the required homozygous recessive genotype at the C locus. So we can move on through the pathway. We do successfully make scales. But we see here that having this homozygous recessive genotype at the S locus prevents us from moving on from regular scales to camouflage scales. So while we did successfully pass this checkpoint, we got stuck right here. So this snake will have regular scales. And so we can once again compare these phenotypes that we see from the crosses of our genotype. And we see that 9 plus 3 of them are bare. 9 plus 3 is 12. And then we have 3 that are camo. And then 1 that has regular scales. And so this is the very recognizable ratio of our of a dominant epistasis event that is the result of a cross between 2 true breeding individuals. And the resulting test cross, of course. And there is another kind that we shall visit. There's a 4th kind, but honestly, I never learned it, and I don't think you have to know it. So there's another kind of epistasis called duplicate recessive epistasis. And so let's say that there is a butterfly. Well, let's say that all butterflies start out with no pigment in their wings. And if they have dominant alleles at both loci, assuming that they have 2 loci that code for pigmenting the wing spots, if they are dominant at both loci, then they successfully deposit pigment into their wing spots. But if they have either, oops, let's not do green. If they have either That was a terrible A. Homozygous recessive genotype at either the A or the B locus. Tire that. It will stop this from happening and no pigment will be deposited. So having either A or B loci, as homozygous recessive will stop this in its tracks. So duplicate recessive, you have to get through 2 loci as being dominant to not be stopped along this pathway in an epistatic fashion. So obviously, this is also called duplicate because both loci are epistatic to the other. Because, if you have lowercase a, and a capital B, this is epistatic to this because it will stop the pathway. But if you have this is epistatic to this, and it will stop the pathway. And so let's revisit our cross. We have 2 true breeders. So we have an A, and a B. B crosses with A, B, B. So we have P, F1, F2. F1 is obviously all F1 individuals are going to be heterozygous. At both loci. And so if you cross 2 of these, you get, or we can investigate the ratio, and we know this. Sorry. Dominant at both, dominant at one but not the other, recessive at one and not the other, and then recessive at both. I never draw letters the same way twice. It's weird. So we can look here, and if we see if there's a homozygous recessive genotype at either locus, that is going to stop the pathway, and we will no longer be able to have pigmented wing spots. So we need to be homozygous dominant at both loci to have pigment. Where'd my blue pen go? Oh, I made it purple, that's right. So homozygous dominant, dominant at both loci. So we have pigment. Here we have a homozygous recessive. It's only at one locus, but it is epistatic to this one. And so we will have no pigment. Oh, same here. No pigment. And both of these loci are homozygous recessive, so no pigment. So once again, we can group up similar phenotypes. We see that these 3 come together. 3 plus 3 plus 1 is 7. And so our 9 to 3 to 3 to 1 becomes 9 to 7 in the face of duplicate recessive epistasis. All right. And so with all this under our belt, it would probably be wise to remember these ratios at the least. 9 to 7 belongs to duplicate because, having a homozygous recessive genotype at either locus will automatically stop the pathway and you'll be stuck right here. So 9 to 7. And then in the case of recessive epistasis, we have our 9 to 3 to 4. And then in the case of our dominant epistasis, it was 12 to 3 to 1 because the dominant allele takes over. So with all that in mind, let's try a problem. Feel free to read this and pause it, try it yourself, and then we'll come back. Okay, so let me get all this nonsense out of the way. Sorry. Okay. We have, which color should we use? Let's use, okay. So in a certain species of plant, flowers occur in 3 colors, blue, pink, and white. A pure breeding pink plant is mated with a pure breeding white plant. All of the F1 are blue. When the blue F1 plants are selfed, the F2 plants occur in the ratio 9:3:4. The genotype that it produces white is lowercase w, but the presence of a single white allele allows pink or blue color to occur. The alleles at the hypostatic locus are blue if they're capital and pink if they're lowercase. What is the genotype of the pink parent? Okay, so how would I answer this question? Immediately, I see this and I know what I'm dealing with. This is going to be an example of recessive epistasis. That gives our 9:3:4 ratio. And so the question asks, what is the genotype of the pink parent? So we see that it's written in the question, a pure breeding pink plant is mated with a pure breeding white plant. So in order to be white, actually, let's keep reading the question. So the genotype that produces white is WW. So if it's white, oh yeah, the presence of a single capital W allele allows pink or blue color to occur. And if we think back to our recessive epistasis example of the flowers, funny that, we remember that it was the D locus, the pigment deposition locus, that was epistatic to the color locus. And the same thing is happening here, except the presence of a single capital W allows for the color locus to come into play. But if you have recessive, if you're homozygous recessive at this locus, then that means you don't deposit color and you are white. So I'm going to say that this W locus is the epistatic locus, epistatic gene, and from this we can tell that if one parent is pure breeding in white, well, in order to be white, they obviously have to have, let me get the eraser. In order to be white, they need to have lowercase w, lowercase w. Actually, we don't even need to deal with that. We can go back to the first parent. So if they're true breeding, they're, pure breeding pink plant. So in order to be pink, in order to have color, remember, the color locus is hypostatic to the pigment deposition locus, this white locus. So we know that we're going to be dealing with some kind of B and then our W's. So if it's pink, that means it's not lowercase lowercase, because if it were lowercase lowercase, then it would fail to deposit the pigment. And so it has to have at least one capital W. And if it has, or that actually helps us to cross out these two answers, because these two would be white due to the epistatic locus affecting the hypostatic locus. And because we know that it's pure breeding, sorry, that got cut off by the webcam. We know it's pure breeding, so it's obviously not going to be heterozygous. Instead, it is going to be homozygous dominant, and that gets rid of these two, because these are heterozygous, heterozygous, this is homozygous. So answer is A. And obviously, we could even ignore that, see that it's pink, obviously, it's pure breeding and it's pink. And we know that pink is recessive to blue at the color locus. And so pink has to be lowercase b, which would cancel out this and this and this. So we get to choose from A and B, and we know that it's pure breeding. So obviously it's not going to be this heterozygote. So we could have, even without working through what recessive epistasis is, again, just read the question and be able to find out for ourselves. I'm going to grab some water really fast, sorry. Right. Did I unmute myself? Sick. Okay. Moving on to the next question. So, scorpions were the first animals to colonize land approximately 440 million years ago. That's actually, well, I think that's been corroborated. It used to be believed that the polypodia were the first ones. You know, people like centipedes, but scorpions are cooler. And so we stand scorpions colonizing land, and the molecular, or maybe not molecular, but fossil evidence corroborates this. Anyway, in scorpions, having more than one stinger has been discovered to be regulated by two genes via dominant epistasis. The dominant U allele builds a second stinger, which is dominant to the lowercase u allele, which destroys these molecules that are used to build the second stinger. And then we have the dominant V allele that blocks the growth of a third stinger, which is dominant to the lowercase v allele that does successfully build a third stinger. So we see right here, right away, that where'd it go? We are dealing with dominant epistasis. And so we're dealing with dominant epistasis. What do we know? What's the first thing you think of? The first thing I think of is that, exactly, we're going to be seeing a 12 to 3 to 1 ratio, because any, or the presence of any, or actually I should use U and V. Oh wait, I just undid it, goodness gracious. So any dominant allele, oh my God, I should not have done U and V because they look so similar. The presence of any dominant allele is going to force a phenotype. And so we can look at these. And okay, so we can even draw our nice little kind of flow chart. I like that. So we have our little scorpion, got a stinger. I shouldn't do that because then I have to draw 8 legs. So 1, 2, 3, 4, whatever, doesn't matter. So we have our guy, got his pinchers, and he has just one stinger, but he can have 2 stingers, and he could even have 3 stingers. I'm going to draw it to make it look even more cool. Wow. Okay. So let's see. The capital U allele successfully builds a second stinger. Let's get another color. So the capital U allele successfully builds another stinger. I think I also mixed this one up. So let's say that this is going to be a second stinger, this second, this third, and that's obviously pertaining to the third. So if we have a capital V allele, we see that this is going to block the growth of a second stinger, as opposed to a recessive V allele, which does successfully build a second stinger. So having lowercase v's will allow us to continue along the chain, but having an uppercase V is going to block and stop us in our tracks. And then if we have a capital U allele, it will, that's U, it will allow the building of even a third stinger after we have successfully built a second stinger. But having a homozygous recessive genotype at this U locus is going to stop us in our tracks and block us from moving on to 3 stingers. And so we can, once again, create our cross. Well, actually, we know what the cross is going to be. 12 individuals are going to have a exactly dominant allele at the V locus, and therefore be limited to a single stinger. I'm going to stop switching colors. 1 stinger. 3 individuals are going to have, or are going to be recessive at this locus, but be dominant. Or that's supposed to be U. Oh God, I even mixed up the letters. It's fine. Oh no, I just mixed up the letters right here. So I'm going to, for the sake of consistency, fix this. Very cool. Okay, so V and then have dominant allele at the second locus, and I can't even draw a single underline. And then for 1, we are going to have homozygous dominant or recessive at both loci. So if we have a single dominant V allele, at least, then we are going to be stopped right here in our tracks. We're going to be stuck at 1 stinger. But if we have a homozygous recessive genotype here at this V locus, then we get to move on. We get to proceed through the pathway, and we can have at least 2 stingers. So now we can check this locus, and we see that we've made it past here. So can we move on from 2 to 3 stingers? We can do that if we have a dominant U allele, as we see in the question. Dominant U builds a 3rd stinger, and so we see that we're not stuck at the V locus, this being epistatic to this, and we see that we can have 3 stingers. And here we see that, once again, we are not stuck at the V locus because the epistatic gene is showing the phenotype, or excuse me, genotype that will allow us to proceed through this pathway. And we also see, or moving on to the hypostatic locus, we see, the genotype that does block this pathway. So we did proceed through here, but we're stopped here. So we're stuck at 2 stingers. So we have our 12 to 3 to 1 ratio. It wants to know how many will have a 3rd stinger. So we should be looking at this, and this is obviously just 3 individuals are going to have 3 stingers. All out of 12 plus 3 plus 1 is 16. So 3/16 is our ratio. And we can apply this to the total. So this is our ratio. This is going to be some fraction. And we know that there are going to be 28 individuals produced in the F2 generation, which is where we see this ratio. or this distribution of genotypes. And so if there are 28 of them, 28 F2 individuals, sorry, I did it again, 28 F2 individuals, 3/16 of them are going to have 3 stingers. And so we can multiply this 28 by 3 divided by 16, and I'll do the math really fast. I should have built this question better because the answer is 5.25. Oh, it was going to work out well, but I accidentally messed it up. So 5.25 of the 28 scorpion nymphs, which are little baby scorpions, are going to have 3 stingers. And that is all I have for epistasis. There are some better examples that I have if you would like them. And I will see you in the, oh yes, dominances. Oh goodness. Zoom, please let me go.